Hello, and welcome to this webinar organized by microLife. My name is Regina Henge, and I'm a professor of microbiology at Humboldt University, and I'm going to chair this session. So our topic today are a second messenger, nucleotide second messengers in bacteria. And there is actually a thematic issue of microLife that will tell you the state of the art of this research in the current issues. There's a link on the journal, and you can get all these articles as a package, basically. And this thematic issue on second messengers in bacteria actually goes back to a 7-year-old or long research program that has been funded by the German Research Foundation, DFG, and where more than 20 people or more than 20 groups in Germany have done research on this topic. So this thematic issue is basically our final report. And as I said, it gives you the state of the art of this issue. Now, what is a second messenger? In principle, a second messenger is an intracellular molecule. In most cases, it's a nucleotide, sometimes a single or a dinucleotide. Sometimes they are also cyclic. But is really important is that second messengers are informational molecules. That means they stand for something else. So let's say a condition outside the cell or something that changes inside the cell. And so they are representative of this change. And they inform the cell to actually mount a certain response that is to react appropriately. And that means so that the cells can adapt. So basically, second messengers are right in the middle of signal transduction pathways between sensing and generating a certain appropriate response. So they're really essential. And their production and their degradation is actually highly dynamic. So there are enzymes, at least one enzyme that makes a certain messenger in response to a certain incoming signal. And then there are also enzymes that can degrade it. And it's the balance between these enzymes, basically, that determines the level of the second messenger. And then, of course, the second messenger is sensed by some other component in the cell that we call an effector. And that actually triggers an output response mounted by a certain target component. And to actually produce an adaptive response. So basically, there are two principles which are important. The first is representation, and that's what I've already been talking about. The second is specificity. So because a certain signal has to generate a certain output reaction, and in the middle is the second messenger, which is a diffusible molecule. But this is actually guaranteed by molecular interaction and recognition. And as you're going to see, in many cases, it's not that simple. But it's even possible that bacteria have parallel acting responses that react to different signals and generate different outputs, but still use the same second messenger, even though this is a diffusible molecule. And this is kind of a paradox, but actually cells manage to generate this kind of specificity, and you're going to hear in one of our talks. Now, historically, the first second messenger that was discovered was cyclic AMP. And it was in bacteria, it was first discovered in E. coli, which was very fortunate because this is, in a nutshell, the most simple system you can think of. It's just one enzyme that makes it, one enzyme that breaks it, and one protein that can sense it. And that's all, basically. So that was very good for the beginning. But actually, in parallel, another molecule, ppGpp, was also discovered, and which over the years turned out to be much more complex. And also cyclic AMP signaling in other bacteria is more complex. But as a general rule, cyclic AMP is usually involved in sensing and reacting and determining the status of carbon metabolism, whereas ppGpp has a much more general role. Basically, it links the nutrient availability to appropriate responses, which go from growth, if nutrient signaling or neutral availability is great, to non-growth and survival. So it's a general stress signal molecule, basically. And then actually, another molecule was discovered, which is c-di-GMP. And this is the one that really made it clear that second messenger signaling in bacteria is much more complex, because about 20 years ago, it was discovered that most bacteria that use c-di-GMP, and that's almost all of them, that they have multiple enzymes that can produce it and degrade it. So why and how and what does it mean for specificity was an important question. A few years later then, c-di-GMP was discovered, and it was, over the years, it was found that it's mainly involved in osmobalancing and linking osmobalance to associated central metabolic reactions. And during recent years, actually, a number of additional second messenger molecules were discovered, some of them very specific, some of them actually playing an important role in phage resistance systems, which we're not going to cover in this session now here. But if you're interested in that, I can refer you to the review that we have written in that thematic issue. So it's Henge and several other authors, including the ones that who are talking here. And there this topic is also covered. So let's get to the speaker. So our first speaker today will be Natalia Joffrey. Natalia, here she is. She is a professor of microbiology at Hanover University in Germany. And she is a leading expert on Streptomyces molecular biology. And Streptomyces is a very interesting bacteria because it has a very complex and interesting life cycle. And as you're going to see, especially c-di-GMP plays a key role in controlling this life cycle. But in addition, Streptomyces uses also c-di-GMP for osmobalancing and other purposes. And this is actually connected. So Natalia will give you a very broad picture of the physiology and how this is linked to molecular regulation. And then we're going to proceed to Eike Junkermeier, who is also here. And Eike is a graduate student in my group, and he works on the specificity problem of parallel signaling by cyclic di-GMP, which he does in E. coli. So this is the topic of his talk. And the third talk will be given by Jörg Stülke. Jörg is a professor at Göttingen University. And he is an aficionado of Bacillus subtilis, so gram-positive soil bacteria. And Jörg is one of the leaders in the field of cyclic di-GMP and discovered many of the important functions of cyclic di-GMP in Bacillus subtilis. And the final talk will be given by Kürşad Turgay, who is a professor both at Hanover University and also at the Max Planck Unit for the Science of Pathogens in Berlin. And in recent years, Kürşad has been working a lot on ppGpp and its role in the general stress response, and in particular in thermotolerance in Bacillus subtilis. So these are our four speakers. And without further using up your time, I would like to give the stage to Natalia, please.
So hello, everybody. Thank you, Regina, for this very nice introduction into this important topic and the event today. And thanks everyone for joining. So we are many, that's very nice. And yeah, so now it's my pleasure to present you some of our key discoveries about cyclic di-GMP and cyclic di-GMP signaling in Streptomyces. So Streptomyces are gram-positive actinobacteria, and these bacteria are very important members of the soil microbiome. And soil is actually quite harsh environment to live in. So they experience here many challenges and develop many different strategies to adapt and to cope with these challenges. So soil is, for example, a very nutrient-poor environment. But Streptomyces can survive prolonged periods of starvation by simply switching into the existence as spores. It is a very competitive environment, and this is one of the reasons, at least this is what we believe, why these bacteria make so many different antibiotics, so to kill the competitors. And of course, these molecules are very valuable for the medicine and for the biotechnology. And soil is actually also an environment that where changes happen quite often and rapidly, especially in osmolarity, because soil is exposed to weather changes, so to rainfall and drought. And Streptomyces evolved many strategies to adapt to these changes in osmolarity. And in all these key processes, or in all these processes, these two molecules that I will be talking about today, cyclic di-GMP and cyclic di-AMP, play a key role in controlling cellular response and survival. So in the first few slides, I will start with cyclic di-AMP and give you an overview about what we know about the role of this molecule in Streptomyces, and I will then switch to cyclic di-GMP. So when we started to work on cyclic di-AMP, the only certainty we had was that actinobacteria, including Streptomyces, have exactly one so-called diadenylyl cyclase domain-containing protein, which is called this A. And at that time, we knew from work in Bacillus that this A is capable of c-di-AMP production, so it converts ATP to c-di-AMP. But we had no idea how these bacteria actually terminate c-di-AMP mediated signaling, because at that time, there was no actually known phosphodiesterase. So the two enzymes that were known from other bacteria, they are not present in Streptomyces. And we also had no idea what are the targets of this molecule and which functions cyclic di-AMP actually controls in Streptomyces. So by simply screening proteins that were predicted to have some sort of phosphodiesterase activity, we identified an enzyme that we renamed to ATIC. And this enzyme actually is a very specific phosphodiesterase for c-di-AMP and hydrolyzes the cyclic dinucleotides to AMP. And once we had the two enzymes, we started to look into phenotypes, and we learned that similar to what we know from firmicutes, and this will be explained in more detail by Jörg Stürke, in Streptomyces, c-di-AMP is also involved in osmostress regulation, because this A mutant, which has low levels of cyclic di-AMP, has troubles in growing and surviving in presence of high salt on nutrient agar. On the other hand, when we looked at the phenotypes caused by high c-di-AMP, so we looked at the ATIC mutant, so this strain has high levels of this molecule, we realized that high c-di-AMP is somehow connected to defects in development. So normally when Streptomyces complete the life cycle, they produce such beautiful spore chains. But as you can see here, the ATIC mutant produces non-differentiated and very flat and sick appearing hyphae. And this strain also fails to produce the typical spore pigment that Streptomyces normally produce. So at the moment, we don't have a detailed mechanistic understanding how c-di-AMP actually contributes to salt resistance and in how it controls development in these bacteria. But we have identified one c-di-AMP target protein. So this is an RCKC domain-containing protein that we call CPA. So CPA binds c-di-AMP, and upon binding of the ligand, this protein interacts with a transporter that is predicted to be a cation-proton antiporter. And studies from the lab from Ray Elliott have shown that c-di-AMP also binds to a so-called YDO-like riboswitch, which is located in the 5' UTR of a gene which encodes a cell wall hydrolase. And we also know that most of the Streptomyces strains contain other or additional YDO-like riboswitches, which also likely controls genes that encode cell wall hydrolases. So this is basically the context that we know about now, that c-di-AMP controls ion transport and cell wall modulating enzymes. And at the moment, we are trying to find additional targets and try to understand the mechanism behind the function of c-di-AMP. And this is the summary where we are now with our knowledge in this area. And now I would like to switch to c-di-GMP. And the set of the enzymes that control c-di-GMP is far more complex than the one that we know from c-di-AMP metabolism. So most of Streptomyces strain in average have 10 of these enzymes, so which have either GGDF or an EL domain. And our lab model, which is Streptomyces venezuela, has 10 of these enzymes, and we know that 4 of these proteins are important for developmental regulation because deletion of RMDA and RMDB, which are active phosphodiesterases, or c-di-GP and c-di-GC, which are active diglide cyclases, this affects or has a consequence on developmental regulation. And at the moment, we know that high cyclic di-GMP blocks differentiation, and this is nicely visualized here in this image. So this is how Streptomyces wild type, so Venezuela wild type looks at the end of the, so when the spores mature and are formed on these aerial hyphae and the colony becomes green. However, in comparison, a strain in which I overexpressed a diglide cyclase leading to high c-di-GMP, this strain is heavily blocked in sporulation. So here we just see vegetative hyphae, which are covered in some sort of matrix material, and this strain cannot go further in the development. On the other hand, a strain in which I overexpressed a phosphodiesterase leading to low cyclic di-GMP, this strain sporulates like crazy. So this strain even skips one developmental stage. So the spores are not formed on these aerial hyphae, but rather so that they are formed as a mass on the agar plate. Yeah. Which I also want to mention here is that cyclic di-GMP not only controls development, but also antibiotic biosynthesis in these bacteria. So our model, for example, produces chloramphenicol. And when we look at the chloramphenicol production in a strain with high c-di-GMP, you can see that this strain produces much more chloramphenicol than the wild type. So it's like 25-fold more chloramphenicol after 7 days of incubation, which means that this signaling molecule can also be used as a switch-on device for antibiotic biosynthesis. But in the following slides, I would like to elaborate on the mechanism. So our picture from today, what we know about how cyclic di-GMP actually inhibits the transition from multicellular filaments to unicellular spores in these bacteria. And using the cyclic di-GMP capture compound, I have identified a developmental master regulator, a protein called BOLD, as a cyclic di-GMP effector. So we see here the structure of this protein in complex with DNA and cyclic di-GMP. And this is a very unique example in biology because BOLD binds actually a tetrameric form of c-di-GMP, so 4 molecules of c-di-GMP. And this is the C-terminal domain of the protein, and this is the N-terminal domain, the DNA binding domain. And actually, when BOLD binds c-di-GMP, it dimerizes, and when it is a dimer, it becomes active. And BOLD is a repressor of developmental genes. So at high c-di-GMP, BOLD dimerizes and represses the expression of genes needed for mycelium formation and sporulation, so that Streptomyces are arrested in this vegetative phase. So in the second transition phase, so in the mycelium formation phase, high c-di-GMP also blocks differentiation, so the transition from aerial hyphae to spores, but by a completely different mechanism. So here a sigma factor called YG is inhibited, and Kelly Gallagher, a postdoc in the lab of Mark Butner, has discovered that a dimeric form of c-di-GMP stabilizes a complex between an anti-sigma factor called RSIG and the sigma factor YG. And because of this, the sigma factor becomes inaccessible to the RNA polymerase, and therefore, under such conditions, late sporulation genes cannot be transcribed and the cells remain caught in this stage. Okay, so knowing that high c-di-GMP activates BOLD and leads to sequestration of YG, we would assume that at a certain time point, the BOLD dimer dissociates and the YG, the sigma factor, becomes free so that developmental genes can be transcribed normally and the developmental program can proceed. And if we think about this model and also consider cyclic di-GMP levels in the cell, we would expect that when the bacteria decides to sporulate, the levels of the molecule has to drop. So we quantified c-di-GMP in the cell, and this is indeed, so this is what we see, and this is kind of in line with our model. So in the vegetative phase, the levels are elevated to repress sporulation, but then there's a drop of the signaling molecule to allow the sporation program to be initiated. But then we were surprised to see that in the late developmental stage, cyclic di-GMP levels rise again. And initially we believed maybe this is just a resetting program, let's say, because the cycle starts with the spore and ends with the spore. So the beginning at the end actually have to be the same. But we also considered that maybe there's an additional effector protein in this late developmental stage, which senses the second increase of cyclic di-GMP. Again, using the cyclic di-GMP capture compound, we identified a glycogen debranching enzyme, which is called GlycX, as a cyclic di-GMP effector, which is particularly important in the late developmental stage because this protein accumulates during sporulation. And here we see the structure of a GlycX dimer. So this is one monomer and this is the other one, and it is an anti-parallel head-to-tail dimer. And you see that two molecules of c-di-GMP are bound here at the end, or here at these areas of the dimer. And the molecule actually stabilizes the dimer and activates the enzyme. So, and here is just a thin layer chromatography results, so where we used purified GlycX in presence and absence of c-di-GMP and glycogen as a substrate. And you can see that c-di-GMP activates this enzyme to degrade glycogen. And GlycX is also a very important enzyme in the glycogen metabolism in vivo. So this is quantification of glycogen, for example, in the mutant, and you see that glycogen accumulates, or we can detect increased levels in the mutant. However, in a strain where we overexpress GlycX, glycogen can be hardly detected. And it looks like glycogen is somehow connected to normal development, because a strain with uncontrolled degradation of glycogen due to overexpression of GlycX, this strain has a developmental defect, so it does not sporulate as fast or as normal or in the same speed as the wild type. And this strain also produces less viable spores. Okay, so I'm already at the end, and we would like to give you a little summary about the c-di-GMP part. So what we know is that Streptomyces use 3 different c-di-GMP effector proteins, which control sporulation and differentiation associated functions in a specific aura. And this aura is actually determined by the cyclic di-GMP levels in the cell and by the affinities of these proteins for c-di-GMP. So in the early stage, so during the vegetative growth, high c-di-GMP activates BOL-D, so the molecule binds to BOL-D, the BOL-D dimerizes and becomes active as a repressor. And also this cyclic di-GMP in this stage favors sequestration of YG. So these molecules, so YG is inactive and inaccessible for the RNA polymerase. But then the cyclic di-GMP levels drop and BOL-D dissociates, becomes inactive, and YG can be released and become part of the RNA polymerase holocomplex and initiate gene transcription. And this aura is supported by the fact that BOL-D has actually a lower affinity for c-di-GMP than the RSIG-YG complex. And in the later stages, the GlycX protein accumulates, and this second increase of c-di-GMP activates this enzyme to degrade glycogen in order to give access to the bacteria, to the stored glucose in this polymer. Okay, so at the end, I would like to thank my great team at the Leibniz University in Hanover, and also all the wonderful collaborators, and ERC and DFG for funding, and of course, all you for your attention and interest to join us here.
Thank you very much, Natalia, for this beautiful talk, which really gave a summary of, I know, so many years of hard work in a very nicely understandable way. Thank you so much. And before we switch to the next talk, I would like to mention, because I forgot that in the beginning, if for any questions you have, please type them in the question and answer session. So we're going to collect these questions, and after the 4 talks, each of them will be 15 minutes, we're going to have a 30-minute joint discussion session where we're going to respond to your questions. Thank you very much. So our next speaker will be Eike Junkermeier, and he is now focusing on cyclic di-GMP, and he talks about local signaling and that it enhances output specificity of cyclic di-GMP signaling. So the floor is yours, Eike.
Yeah, thank you. Hello, everybody. I'm going to talk about the topic of the review which we contributed about local signaling in cyclic di-GMP signaling networks. So, in a very simple signaling model, the intracellular concentration of the second messenger is controlled via the antagonistic activity of a single cyclase and a single phosphodiesterase. And upon reaching a certain concentration within the cell, cyclic di-GMP binds to specific effector components and thereby activates or sometimes deactivates specific receptors and creates an output reaction. However, there are a couple of observations which do not align well with this standard model of cyclic di-GMP signaling. The most striking one is that one can find a multitude of the enzymes involved in making and breaking cyclic di-GMP encoded in the chromosomes of most bacterial species. Just to give you some numbers, we've heard this already from Natalia. On average, one can find 10 diagonal cyclases encoded in the chromosome of a single species, but the numbers can go up to 50, and the same goes for phosphodiesterases. In some model organisms, we know that these DGC and PDE encoding genes are not only expressed, but they are present and active at the same time within the cell. Also, quite frequently, there have been in the literature reports of specific phenotypes of knockout mutations of particular cyclases or phosphodiesterases, which often involved direct protein-protein interactions. Also, in E. coli, we have a really interesting finding, which is that E. coli maintains basically at all time points along its growth really low intracellular cyclic di-GMP concentrations of around, or which never really exceed 150 nanomolar within the cell. There is, however, one exception, which is a really specific layer within the biofilm, but in general, one can say that E. coli does not really increase its intracellular cyclic di-GMP concentration to activate the effector or target systems present in the cell, although one can clearly see an output from these effector components. Also, quite strikingly, in many cases, there are multiple of these effector components present at the same time within the cell. So the key question is here, how can multiple effector or target systems be controlled in a specific manner if all of them use the same diffusible second messenger in the cytosol? One explanation which has been proposed already many years ago is the formation of so-called local signaling modules. And the idea here is quite straightforward, which is that specific cyclases or phosphodiesterases do directly team up with really specific effector or target systems in a multi-protein complex to form a so-called signaling module. Within this signaling module now, cyclic di-GMP gets produced right next to its receptor binding site. And based on the findings which I've shown on the previous slide, we define 3 criteria which have to be met to conclusively demonstrate such a case of local signaling within the lab. So within such a locally controlled module, one can find really specific phenotypes for single or specific cyclases or phosphodiesterases, which do phenocopy the phenotypes of the respective effector or target systems which are controlled within this module. Also, as I've already mentioned, there are direct protein-protein interactions involved. So the specificity between the cyclase and the effector-target system is facilitated by direct protein-protein interactions. And also under conditions where one can clearly see an output reaction, meaning a phenotype, the intracellular cyclic di-GMP levels can be significantly lower than the effector binding or the binding affinities of the effectors. Or the intracellular concentrations, they do remain unchanged in active site mutants of cyclases or phosphodiesterases, which, however, do show a clear phenotype. In the case of E. coli, these strikingly low intracellular concentrations are produced by the activity of one strongly active so-called master phosphodiesterase called PDH. And PDH is, in the case of E. coli, constantly acting within the cytosol or by degrading cyclic di-GMP and maintaining these really low levels. And these really low levels in the case of E. coli are also a prerequisite for these local signaling modules to act on a local level. Now, the idea for the talk now is that I would like to guide you through some examples of such locally controlled systems, and we will see if we can apply these criteria to these systems. Most prominent one, or the best studied one, might be the enterobacterial cellulose secretion system. And as most of you know, the secretion and also a modification of cellulose is allosterically activated by cyclic di-GMP in E. coli and also many other bacteria. Cellulose in E. coli is an exopolysaccharide, which is secreted and is part of the biofilm matrix of E. coli. And together with amyloid curly fibers, it forms the biofilm matrix. Here you can see such a model which one can use in the lab to study E. coli biofilms. This is a strain which is producing both components, curly and cellulose. And you can see here that this strain was incubated for 5 days on LB agar plates, which contains a certain red dye, which stains curly and cellulose. And as you can see here, these two components do form a composite material, which in which these cells are embedded, and it has some really interesting patterns of a buckling or folding up and wrinkling colony. And it also has some interesting tissue or almost tissue-like properties. Now, in a variant which is not able to produce cellulose, which can be, for example, achieved by knocking out BCSA, the glycosytransferase, or the catalytic core, basically, of the cellulose secretion machinery, one ends up with a macrocolony which only consists of the amyloid curly fibers, and they look clearly different from the strain producing both curly and cellulose. Here you can only see a pattern of concentric rings. Now, the really interesting finding was that between the 12 diagonal cyclases of E. coli, it is specifically just the knockout of one specific cyclase, DGCC, which phenocopies this cellulose-free macrocolony morphology. Also, I don't know if you can see it on the screens, a knockout of one specific cyclase, PDEK, has been found to slightly increase the cellulose production. Maybe if you don't see it here, you just have to trust me. And these knockout mutations in DGCC and PDEK, they do correspond also to active site mutations in the cyclase or phosphodiesterase domains of DGCC and PDEK. Now, to make this long story a little bit shorter, DGCC and PDEK have indeed been found to directly interact with the cellulose secretion machinery by interacting with BCSB, which is a component within this or of the cellulose secretion machinery. Here, I've only shown the key components, basically important for this talk. The cellulose secretion machinery is, of course, way more complex. There are also other BCS components involved, but I'm not showing them here. And there are also multiple copies of BCSB. However, DGCC and PDEK, by directly interacting with BCSB, they have been found to function as a local source and sink for cyclic di-GMP in the activation of the cellulose secretion machinery. And this has also been shown in a in silico or in a mathematical modeling approach, which also, to make this long story short, showed that the hitting probability of cyclic di-GMP for its targets, the PZ domain of BCSA and BCSE, drastically increases if the components, the source, sink, and target of cyclic di-GMP are in or upon a distance lower than 10 nanometers. So this, on the one hand, this mathematical modeling approach showed that this signaling principle in principle works, but it also showed that the signaling here works without the formation of a closed microcompartment within the cell. So what this means is that cyclic di-GMP, which is produced locally here, has just in principle two options. It can either bind to the PZ domain of BCSA or BCSE, or it can diffuse away into the cytoplasm where, however, it gets degraded via PDH. Another example of such a locally controlled exopolysaccharide secretion system is the production of another exopolysaccharide by the NFR system, which is also stimulated in a cyclic di-GMP dependent manner. So here, this NFR system, it produces a mannose-based exopolysaccharide. However, also to make this long story short, we found that this exopolysaccharide is not involved in the biofilm formation, which is why we are not looking at E. coli marker colonies here. We only found out that it's, in principle, not involved in the biofilm formation. But interestingly, we found that this exopolysaccharide is used by a certain phage as an initial receptor for adsorption. So instead of looking at macrocolonies, we are looking at the ability of the phages to infect E. coli, which indirectly tells us about the presence of this glycan. Interestingly, also here again, we can see the same principles. So a knockout of one specific cyclase, DGCJ in this case, leads to a plating defect, while none of the other 12 enzymes or DGCs in E. coli showed an effect. And here again, this plating defect could only be restored via the expression of the wild-type DGCJ, but not a catalytically inactive variant. Here we also found that DGCJ indeed directly interacts with NFRB and functions as a local source of cyclic di-GMP in the activation of the system. And here, in principle, the same principles apply, which is that this close vicinity between the source and the target of cyclic di-GMP enable a specific activation of the system. And this also could explain why NFRB, although it has a high affinity for cyclic di-GMP or the mesh end domain of NFRB, why this KD is still 10 times higher than the intracellular concentration within the cell. So at the time points where we clearly see an output of this system. One thing I would also like to point out here is that the protein-protein interaction between the source of cyclic di-GMP and its target has no further regulatory role than just providing the specificity and the vicinity between the source and the sink. There are, however, examples where such protein-protein interaction does have a regulatory function. And such an example is the control of the expression of CSGD. CSGD is a transcriptional regulator in E. coli, which drives the expression of the coli subunits and DGCC, and thereby indirectly controls the activity of the cellular secretion machinery. So CSGD is a key player in the formation of E. coli biofilms. Now, the expression of CSGD is controlled via a module consisting of a phosphodiesterase, PDR, a cyclase, DGCM, and MLRA, which is a transcription factor. It is not a cyclic di-GMP effector component. So MLRA is not binding cyclic di-GMP, but we'll see in a second how this regulation works. Now here, as I've mentioned, PDR, DGCM, and MLRA, they do form or all of these components, they do interact within this module. And as long as PDR is not active as a phosphodiesterase, it reduces or it blocks the DGCM MLRA-driven activation of expression of CSGD. However, if PDR gets active as a so-called trigger enzyme, it releases its inhibitory role on DGCM and MLRA, and thereby enables the expression of CSGD. So here PDR has on the one hand a regulatory role, and it functions as a trigger enzyme it functions as a trigger enzyme and thereby as a cyclic di-GMP effector component, which does not bind cyclic di-GMP, but it degrades cyclic di-GMP. Here again, interestingly, PDR degrades cyclic di-GMP, which is specifically produced by another cyclase called DGCE. With this done, and I would like to sum up what I've shown you today. So I talked about the 3 criteria which one can define for local cyclic IgMP signaling, which are specific knockout phenotypes of specific cyclases or phosphodiesterases, direct protein-protein interactions between cyclases and effector or target systems, and binding affinities which are higher than the intracellular global concentration of cyclic di-GMP. And I've told you today that there are 2 types of local signaling. On the one hand, where the protein-protein interaction just associates a local acting source or sink of cyclic di-GMP to a specific effector or target system. And also another type in which this protein-protein interaction has a regulatory function, exemplified by the trigger PDE, PDR. There are, however, other examples, as you've maybe realized in this talk today. I was only giving examples from E. coli. If you're interested now in this kind of signaling and local signaling, you can find many more examples also from other bacteria in the review. So if you've not checked it out or if you've not read it yet, maybe you are now interested in, for example, another regulatory interaction between a cyclase in the case of Pseudomonas, where the lab system also is controlled on a local level. One thing I would like to highlight in the end is that if there is a case of local signaling within the cell, this does not mean that these cells do only sit in this local signaling mode. So there definitely is a switching from a local to a global, from a local to a global signaling mode. For example, also in E. coli. And something which I've not told you due to the interest of time is that specificity can also be achieved by fine-tuning the global intracellular levels. If you're interested in that, also I can highly recommend to check out the review. And with this, I'm done. I would like to thank, of course, all of the members of our group, foremost, of course, Regina for being my PI, and Ronja for being an awesome lab mate, and all of the members of Regina's lab from the last or the many years in Regina's lab, which did all the work and which I used to, or which I summarized here. So, and also I would like to thank you for your attention.
Thank you very much, Eike, for this beautiful summary of a very difficult topic. We're actually accumulating questions. This is very nice, and we're going to answer them at the end of the talking session. Okay, so let's switch to the next talk that will be given by Jörg Stühlke. And Jörg, are you ready?
Yes.
Okay. Okay, so this is a talk about cyclic di-AMP and why this is actually an essential nucleotide second messenger and what it actually does in Bacillus subtilis. So please go ahead, Jörg.
Okay, thank you very much, Regina, for the introduction, and thank you for the organizers who put together this nice webinar. Well, we are studying cyclic di-AMP signaling in Bacillus subtilis, a gram-positive model bacterium. This molecule has been discovered in 2008 by Gregor Witte, whom you can see here in Munich. And the enzyme is synthesized, the molecule is synthesized by enzymes that are called deadenylate cyclases. Cyclic di-AMP is present in most gram-positive bacteria, in many gram-negatives, including cyanobacteria, also many proteobacteria, but not in enterobacteria like E. coli. And it's even found in some archaea. cyclic di-AMP is essential for firmicutes, and upon accumulation in the cells, it can become toxic. So we dubbed it an essential poison. Cyclic di-AMP can also be secreted from the cell. And you know, many gram-positive bacteria are pathogens like Staphylococcus aureus or Listeria monocytogenes. So we do not, our body does not produce cyclic di-AMP. And if cyclic di-AMP is released by the bacteria, then our immune system recognizes the molecule and knows now we have to deal and to fight an invader. Okay, in Bacillus subtilis, there are actually 3 enzymes that can produce cyclic di-AMP. This A, which has already been briefly introduced by Natalia, which is also present in Streptomyces, c-di-GMP, and the third enzyme, c-di-S, is only active during spirulation in Bacillus. And there are 2 enzymes, 2 phosphodiesterases, which can degrade cyclic di-AMP, and they are shown here. And you see several of these proteins are located in the membrane. Now, the key question and what really triggered my interest in cyclic di-AMP is what makes this molecule essential and also what makes it toxic. And what we thought is that cyclic di-AMP might be essential because it stimulates the activity of an essential enzyme, like, for example, enzymes of cell wall biosynthesis. And alternatively, it could also counteract something that is toxic. And this we know for RNase 3 in E. coli, this is essential because it degrades a toxic RNA molecule. And of course, it could also be a combination of both. But now the problem is that none of the known targets is essential. And also none of the known targets is toxic. So what might be the reason? So one might be that the essential target has escaped detection. The second, that essentiality and toxicity of cyclic di-AMP are the 2 sides of the same coin, and that cyclic di-AMP might control a complex process that is essential but must be limited at the same time. And I will show you that it's exactly the last 2 points that are correct. And what we found, and also others, is that the control of potassium homeostasis is the essential function of cyclic di-AMP in Bacillus subtilis. And for other gram-positives, we can also say it's the control of osmotic homeostasis, and potassium is one of them. So if you look at target molecules of cyclic di-AMP, then you see there are quite a lot of different proteins, and there's even an RNA molecule, a riboswitch. So cyclic di-AMP binds both proteins and RNAs. And what you also can see, there are 2 potassium uptake systems, a potassium export system, and a riboswitch that controls the expression of potassium transporters. And then we have an osmolyte transporters and 2 signal transduction proteins. So one thing that is already interesting is that cyclic di-AMP is the only second messenger that controls the biological process, and this is potassium uptake, by binding an enzyme or transporter in this case, and the riboswitch that controls its expression. So 2 completely different molecules, both have evolved to be controlled by cyclic di-AMP binding. Now, we thought we should have a closer look to potassium and cyclic di-AMP, and it turned out that indeed, at low concentration of potassium in the medium, the cyclic di-AMP concentrations are rather low, and at high concentrations, there's also more cyclic di-AMP. And here we have strains where only one deadenylate cyclase is present, and you see the same result. So we can conclude that potassium availability is the primary signal for which cyclic di-AMP serves as the second messenger. Now, we also have observed that, in fact, it's potassium that can be toxic for cells that lack cyclic di-AMP. So we have two strains, a wild-type strain and a strain lacking cyclic di-AMP. We cultivate them at low potassium concentration. Everything is fine over time. Now, if we have a high potassium concentration, you see after 5 hours, the cells of the strain lacking cyclic di-AMP, they are all ghost cells, so they have died. So potassium is toxic in the absence of cyclic di-AMP. And this brings me to a small model. So if there's a lot of potassium in the medium, there's a lot of cyclic di-AMP, which in fact activates potassium export systems and inhibits potassium uptake systems or their expression, so that the amount of potassium in the cell remains limited. Now, in contrast, if the potassium concentration is low, then there's also a low concentration of cyclic di-AMP in the cell, and then the molecule cannot bind its interacting proteins, and we have an effective uptake of potassium so that the cell get enough potassium in the cell. So we can say that cyclic di-AMP inhibits potentially toxic potassium export uptake, and it's required for the export of potassium ions. Now, let me turn to something different. There are two signal transduction proteins, DAR-A and DAR-B, that don't have any specific activity like the potassium transporter. So these proteins only serve for signal transduction. In the case of DAR-A, it's a so-called P2-like protein. It's the largest class of signal transduction proteins that are particularly important in nitrogen regulation. And DAR-B is a protein composed of so-called CBS domains that are known to bind adenine nucleotides. And we have identified interaction partners of DAR-B, and we found two hot candidates. The REL is the ppGpp synthesizing and degrading enzyme, and ppGA is the pyruvate carboxylase, which replenishes the TCA cycle. And we did an alternative experiment, just a control experiment, where we did a pull down of proteins of Bacillus subtilis using a so-called streptactin column, to which biotinylated proteins can bind. And in Bacillus, there are two biotinylated proteins, so they both bound, of course, to our column, but there was one additional protein, and this was DAR-B. And of course, now, if you see this here, it's obvious that this is most likely mediated via the interaction with the pyruvate carboxylase. So we can conclude that the REL and ppGA proteins are attractive candidates for further study. And we did this, and here we have an interaction study between strep-tagged REL and DAR-B protein. And what you can see here, the interaction takes place in the absence of cyclic di-AMP. We can detect DAR-B, but if cyclic di-AMP is added, there is no DAR-B. So the interaction corresponds to the availability of cyclic di-AMP, and specifically cyclic di-AMP inhibits the binding of DAR-B to REL A. And this has also been shown by ITC experiments. Now I told you that cyclic di-AMP is the second messenger for the availability of potassium. So we had a look at the link between potassium concentration and the interaction between the two proteins. And again, it turned out that the interaction takes place in the absence of potassium or very low potassium concentration, and the interaction is very weak at a high potassium concentration. So this now tells us that Da-A binds to Rel-A in the absence of cyclic di-AMP under conditions of potassium starvation. And this brings me already to the model. So at low potassium concentrations, the apo-Da-B, the Da-B without cyclic di-AMP, binds to Rel. And we have also shown that this triggers ppGpp synthesis and inhibits the hydrolase domain so that the ppGpp concentration in the cell increases. So we can conclude that potassium starvation triggers a stringent response in a c-di-GMP dependent manner. And this makes a lot of sense because potassium is required for many cellular processes like translation in the ribosome. And if there's not enough potassium, then proteins cannot be synthesized. So it makes a lot of sense that then this is perceived like amino acid starvation, just in another molecular way. And I told you, we also saw an interaction between Da-B and the pyruvate carboxylase. And what we have shown before is that potassium starvation requires the synthesis of positively charged amino acids, because potassium is the counter ion for the negative charges of DNA. And if there's no potassium, then the cells needs other positively charged molecules to buffer the negative charge of DNA. And these are positively charged amino acids. And they are made from glutamate, which is derived from the TCA cycle. So that means in the absence of potassium, we have a high demand for glutamate. And this means we have a high demand to replenish, to refill the TCA cycle. And now you see an assay, an enzymatic assay of pyruvate carboxylase in the absence and presence of Da-B. And we can conclude that Da-B, again, the cyclic di-AMP free form, the apoprotein, activates ppA under conditions of potassium starvation. And now what is really interesting is that a link between cyclic di-AMP and pyruvate carboxylase has already been described in Listeria monocytogenes. But there, cyclic di-AMP at high potassium concentration directly binds the pyruvate carboxylase and inhibits the enzyme. And the outcome is that at high potassium concentration, we have a reduced ppA activity. In Bacillus, the mechanism is different. Here, it's potassium starvation, which needs to enhance expression of pyruvate carboxylase and enhanced activity of the enzyme. But the general logic behind this regulation is very similar. We have a link between the extracellular potassium concentration and the intracellular activity of the pyruvate carboxylase. So this shows that even in very closely related organisms, the specific mechanism can differ a bit, but the logic often remains the same. So now to sum up, I can say that the control of potassium homeostasis is the essential function of cyclic di-AMP in subtilis. At low potassium concentrations, the interaction between apo-Da-B and Rel triggers the stringent response. And ApoB also activates the pyruvate carboxylase to refill the TCA cycle. And so we can conclude that cyclic di-AMP really controls several aspects of cellular homeostasis. And these are the people involved in this work. Larissa Krüger, Jan Gundlach were PhD students, Dennis Wicke still is, and Christina is a technical assistant who was involved in this project over the years. And we have excellent lab members and collaboration partners in Göttingen, in Hanover, in Greifswald, Frankfurt, and also in the United States, without whom this work would not be possible. And of course, I'm also really grateful to Regine for putting together this fantastic SPP program, which was funded by the DFG. And with this, I'm at the end and would like to thank you for your interest.
Thank you very much, Jörg, for this beautiful summary and talk. I would like to say this talk also nicely illustrates where the whole field of second messenger signaling is actually moving to. So we are no longer looking at single processes only, but now we look at the connection between the processes. So basically the field moves towards a systemic view of regulation in the cell. So this is really interesting. And you also gave the keyword for the next talk by mentioning the relationship between cyclic di-AMP and ppGpp. So the last talk will be by Kürşat Turgay, and this is about ppGpp, again in Bacillus subtilis, and now this is about the heat shock response. And I should say that ppGpp has been known for a long time. It has mainly been studied in E. coli, and it was known to bind to RNA polymerase. But in recent years, and especially in Bacillus subtilis, so many new functions and above all mechanisms have been discovered in Bacillus subtilis. So this is an example here. And so, Kürşat, you can start.
Okay, so I will go on after the nice introduction from Jörg. And the general theme of stress response in ppGpp was already highlighted by Jörg. And we are more interested in, or my lab works more on the heat shock response in Bacillus subtilis, where we also look, as Regine already said, in processes like thermotolerance and thermoresistance. And heat shock response is all about, at least what we look on, is proteins. And proteins have to get folded to be functional. And upon protein folding stress, like heat, proteins can unfold and misfold, and then they can form protein aggregates, which in general is bad for the cell, and it helps or induces that they cannot function very well anymore. And here I want to show you an experiment where we took the subtilis cells, grew them at 37 degrees where they can grow perfectly, and then we added a heat stress or exposed them to a heat stress which is very high, 53 degrees in this case. And then you can see that the B subtilis cells are dying. So they cannot respond strong enough against that heat stress. But if we put up a little pre-shock at 48 degrees, which is heat, but can be stand by the cells very easily, then what we see is that almost all the cells survive the otherwise toxic heat stress at 53 degrees. So by putting a little heat shock at the beginning, we kind of prime the cells. They induce a kind of response to the heat shock, which then allows the cells to survive an otherwise toxic heat shock. And here you can see an experiment where we just looked at the proteins and prepared the protein aggregates, the pellets here. And you can see if we have the 53 degree stress, then the cells produce a lot of protein aggregates. And when we have the 48 degree pre-shock before the 53 degrees, then we don't see a lot of aggregates. And we can also detect these aggregates by fluorescence microscopy with a marker like the small heat shock marker or here in the GFP. And we can also see this dark phase in cells under normal microscopy. Okay, so what are the processes which go against this, and this is called the protein quality control system or the heat shock response. And one important heat shock response is regulated by a repressor called HRCA, and HRCA is a repressor of an operon or of different operons which encode chaperones like GROEL or DNAK. And it's a repressor, and when heat is going up, HRCA is aggregating, and the chaperones are induced. Interestingly, one of the chaperones, GROEL, can then activate HRCA again and turn off the system. But once there's a lot of stress, the GROEL titrates away to the aggregated proteins, and thereby the system is always on and running unless enough chaperones are made, and then the whole system is turned off again. Another important system is, besides the chaperones, which can repair and refold misfolded proteins and also inhibiting aggregation, is a system where there's a protease which can recognize, helped by adapter proteins and kinases MCSB and MCSA, which then can target the transcription factor, which can also be inactivated just by heat. And thereby, again, these protease complexes are made. They can target unfolded and misfolded protein or protein aggregates, which can get unfolded and then also degraded. And this is all controlled by this transcription factor, or again, a repressor CTSR, which is then targeted also to ppGpp for degradation. And thereby, we can also control in a similar mechanism of the titration model the activity of this protease, which can then remove protein aggregates and can also repair misfolded proteins. Okay, there's another system called sig-B, which is like the general stress sigma factor in Bacillus, which encodes also many genes, and this is all controlled by sigma B is a sigma factor that's controlled by anti-sigma factors and anti-sigma factors, which can also sense environmental stress or energy stress, which can also prepare the cells during heat shock response against stresses which come from the environment and from the outside. And the last but not least system is also a heat shock response, or actually also an oxidative stress response, which is mediated also by a transcription factor, an unusual transcription factor, SPX. SPX is usually targeted by an adapter protein to another protease system, ClipXP, and is getting degraded there. But when there's heat or oxidative stress, this adapter protein goes to aggregates and cannot target SPX anymore. And then SPX is active. It interacts with the alpha CTD of an RNA polymerase, and it can turn on the SPX regulons, which are chaperones and thyroidoxines, but it can also inhibit the transcription of proteins which are connected to translation. And here you can also see an experiment that the delta SPX knockout has a thermotolerance phenotype, and you can also see that in a delta SPX, the priming doesn't help, and we get a lot of aggregated proteins, which is not good for the cell and also toxic. And here's an experiment which I like a lot, where we put SPX under the induction of an IPTG-inducible promoter, and then we follow ribosomal RNA. And when we add the IPTG, SPX is induced, and the ribosomal RNA transcription is turned down. And at the same time, the transcription and translation of redox chaperones like thyroidoxin B is turned up. So SPX is really a kind of dual role, that it can induce heat shock and general stress response and the oxidative stress with tricks A and B, but at the same time, it can downregulate translation on the transcriptional level of RNA operons and ribosomal proteins, and transcription of the genes of them. It also has a quite pleiotropic influence on other processes in Bacillus. Okay, and when we look at the transcriptional changes during thermotolerance in Bacillus, then we can clearly see that SPX regulated genes are regulated very highly under these conditions. They are both downregulated, as I just mentioned, and upregulated genes. And the other interesting part, what we saw here, is that many stringent response genes were affected under these conditions, in addition to many translation-regulated genes, which are also downregulated upon the thermotolerance conditions, as heat shock with a pre-shock. Okay, so what is stringent response? We already heard from Jörg and others that we have here ppGpp, which is can be active as pGpp, ppGpp, or ppGpp, and it's an alarmone which is synthesized in Bacillus subtilis by synthetases, ppGpp synthetase. There's first of all REL, which Jörg already mentioned, which has a regulatory domain, a hydrolase, and a synthetase domain. And we also have small synthetases, REL-P and REL-Q, which only has the synthetase domain, which can also synthesize ppGpp. And here's a little scheme where you can see the model on what we think how that system works, is that when we have amino acid starvation, we get a lot of many deacyl tRNAs, and these deacyl tRNAs can interact with REL, and then REL can go to the ribosome and sits on the ribosome, and there its synthetase domain is turned on, and it can make ppGpp, while when it is off the ribosome, most of the time the hydrolase is on, and ppGpp actually get degraded. The small synthetases can also synthesize ppGpp, and we also know of the hydrolases which can change a ppGpp and a pGpp, for example. We know that in our system these are not so important, and what Jörg already just mentioned is this REL protein, which in his model can interact with the star A protein, can form this dimer, and then of course it cannot also not go to the synthetase, to the ribosome and synthesize ppGpp. And so it's a sensing system, so this REL protein can apparently also sense other proteins which by direct interaction with REL then can also control or interfere with or use the ppGpp system. Okay, and normally here's just a little growth curve, and you can see when the Bacillus cells get into stationary phase, ppGpp goes up, and the reason for that is that nutrients are getting scarce. Once nutrients are getting scarce, one way to react to it, especially when you go into stationary phase, is to shut down translation. That not because the cells stop to grow, they stop to differentiate. And that is an important way that it's a kind of break which slows down, for example, translation, but also other processes in these cells. Under other conditions, when there are not enough amino acids, also amino acid synthesis can be turned on. And here you can see a little scheme. So when stringent response in Bacillus is turned on, it can first inhibit replication. It can inhibit translation at many steps because ppGpp can bind to GTPases and inhibit the GTPases with this additional dual phosphate there. And it also interferes with the synthesis of GTP in the cells, and the GTP level can also interfere with the transcriptional regulation. And there are also other proteins called COT-Y. And so therefore, inhibition of purine, and we now also know pyrimidine biosynthesis can appear via ppGpp. The transcription can be reprogrammed to cope with translation. And of course, if there's an amino acid, if there are not enough amino acids there, then the amino acid synthesis can also be turned on in these cells. Okay, so what happens in thermotolerance when we get rid of ppGpp, and we could observe that it didn't have a big influence on thermotolerance or thermoresistance, but we could observe that there were more higher translation, and you saw a stronger formation of aggregates under normal, when you switch to 53 degree under strong stress, which could be alleviated with this pre-stress. But most interestingly, when we made a mutant, we could make different ways. And this is just one example where we deleted REL, which is the only hydrolase, and therefore we get a lot of ppGpp in these cells. And what you can see is that all these cells with high ppGpp level can basically survive this strong heat shock, and only the wild type cells are dying under the 53 heat shock stress. And the other interesting thing is we almost saw no aggregates in these cells. So they have higher ppGpp list, which results in increased thermotolerance, less aggregates, and they seem to appear always stringent. Okay, then we looked at the translation. We could see that in wild type cells, if you just grow them, we can measure translation. And when they go actually to 50 degree, they grow even faster. You see a higher translation. And when we looked at this in ppGpp 0, we even saw another additional increase of translation, especially in the beginning when the 50 degree started the growth. And in the REL mutant, we saw lower translation in general. So ppGpp, when it goes up, can shut down translation. And we also observed that in the REL mutant, we saw here, we look at the ribosome profiles in Bacillus, in cells, and its sucrose gradient, and then you can see the 70S, the 50S, the 30S, and here's the polysomes, which are translating. And when we have a REL mutant, we see much less of a translation. And when we go to the 48, 53, to the thermotolerance conditions, we also see the formation of so-called disomes or 100S ribosomes, which get dimerized. And this dimerization is also depending on ppGpp. And this is a kind of ribosome which is parked because the translation is shut down, but the ribosomes which are not translating can also be put in this form where they can be reactivated when you want to come back later to translation. Okay, so is ppGpp really turned up upon heat shock? And here we can see that at 50 degree, we see an induction of ppGpp, which then goes down again. So it goes down after 10 minutes. And this is an induction of ppGpp by adding norvaline, or here's a delta REL mutant. And the other interesting thing we observed was that under heat shock conditions, we only have this brief, short peak of ppGpp, and we could see in all our results, we did all kind of experiments to test it, that these processes were not affected by the short ppGpp course, but the translation was really strongly affected. And one reason could be, I just showed you that ppGpp is synthesized on the ribosome by REL sitting on the ribosome. So it could represent also a kind of sink or local synthesis, local induction of ppGpp, because only a low percentage of the ribosomes actually can interact with REL, and they synthesize this ppGpp, but around them are many other ribosomes which then can also sense ppGpp. So in a way, it's a little bit like this local synthesis and local sink model we heard from Eike and from Regine, which can happen with like a c-di-GMP, and here in a similar way, we have the activity of ppGpp kind of under heat stress conditions only on the ribosomes. And we also know that only REL is important for the ppGpp synthesis upon heat stress, and the small synthetase don't produce that much more ppGpp because of this. Okay, and then we also were interested because we know that SPX is also downregulating translation, whether there's an interaction between them. And if you look at ppGpp 0, the growth is not really affected. It's almost like wild type at 50 degree. Delta SPX has a growth effect, but the combination of ppGpp 0 and delta SPX have even a stronger growth effect, which kind of argues that these two systems might interact. They appear to be independent, but both can also be involved in the downregulation of translation. So our simple model is in a way that when we have normal grown cells, that translation is up, ribosomal proteins are made, heat shock proteins are down, then the heat shock comes up, it's sensed by the different systems I just introduced, SPX, HRCA, CTSR, and sigma B. The protein quality control capacity is induced because the number of chaperones and redox chaperones are made. But at the same time, ppGpp is induced, and SPX inhibits the transcription of translation-related proteins, and ppGpp on a direct level, on the protein level, post-translation, you can interact with these GTPases in the translation and can thereby inhibit translation. And the inhibition of translation under stress conditions is, of course, very important, because if we wouldn't inhibit the translation under these stress conditions, we would have many more proteins made, but then under the heat shock conditions, they would just aggregate and they wouldn't become functional. So and also for the chaperone systems, you just reduce the load of proteins which could misfold or aggregate, and thereby also allow the chaperone system to work better and more efficiently on the already misfolded and aggregated proteins without getting new proteins synthesized at the same time, which would increase the load and would not help the protein quality control system, the protein homeostasis under these stress conditions. Okay, with this, I'm finished. I would just want to acknowledge Stefanie Runde, who worked a lot on the SPX system. Heinrich worked on SPX and ppGpp. And now we have Christina and Fabian, which also work on heat and ppGpp systems, the Bacillus subtilis. And maybe in another seminar later, I can talk about their work. And Ingo and Regina also work together with Heinrich and help a lot. Okay, with this, I'm ready to take questions.
Okay, thank you, Kürşad, very much for your talk. So you can see how complicated it can get with second messengers. Huh? It started very simple, but now we are really at a very complicated picture. So thanks to all the speakers, we have a number of interesting questions in the talk, in the question and answer session now. We are a bit over time, but we have time for a few questions. So some of these questions are very specific, others are a little more general. So I'd like to start with a bit more general question that is maybe something for Natalia or Eike to answer. So what is generally considered to be high or low concentration of cyclic di-GMP or cyclic di-AMP? So because we always speak in these relative terms, what is high and what is low, and what does it depend upon that we call a concentration high or low?
Okay, great question that I actually answered by typing. Yes, unfortunately in Streptomyces, we don't know the exact concentration of these molecules because these bacteria change the morphology, the volume of the cell, and it's very difficult to calculate. But in Streptomyces, we estimated it to be in the low micromolar range, so let's say up to 5 micromolar, which is already high if we compare it to 150 nanomolar in E. coli. So I would say this is the range we are talking about high and low.
Yeah, maybe I can add something to it because I have also worked on that for a long time. I mean, what really matters is actually the KD of the effector systems. So if you have a concentration which stays way below the KD you would need in order to, or the concentration you would need to activate based on the KD of an effector, then we talk of a low concentration. And a high concentration is basically a concentration that is beyond the KD for certain effectors. So this might be very different in different bacteria. So like in E. coli, it's very low. And what we can consider high in E. coli would still be low in Streptomyces, for instance, because it depends on the KDs of the effector systems. Then I have here another interesting question. This is something for Eike. Does N4-phage target the NFRA directly or other NFRA-associated proteins? Because you have talked about that its primary target is the polysaccharide.
Yeah, so the phage does so this is like a general mechanism which can often be observed for phages to initially basically reversibly bind to a glycan on the cell surface and then find in a second step in most of the cases protein receptor, and this is most likely also the case for the N4 phage. So it binds in the first step most likely the exopolysaccharide secreted by the system, and then in a second step, the NFRA protein. And this has been shown many years ago that it indeed binds to the NFRA protein. So this has been shown in vitro. And we, based on our results, most likely consider that it binds also the exopolysaccharide.
Okay, thank you, Eike. There was actually another question for Natalia, but it seems that Natalia has answered the question, so they have disappeared. But I think that might be interesting for a general audience as well. And there was a question about ClickX. So whether this is general, that ClickX is targeted by c-di-GMP, or can you recognize that at the molecule in other Streptomyces, etc.?
Yes, that was a question from Khaled, who is here. So, yes, so we actually purified ClickX from E. coli, which also has it, right? But it does not have the binding signature, and it also does not bind c-di-GMP. So this is what we tested. But some Streptomyces have a second copy of ClickX, which does not have the motif. But some of them are not even expressed. So there are these two types of ClickX with the motif and reaction or binding of c-di-GMP and without. And all the Streptomyces strains that we checked have one version of the protein with the c-di-GMP binding signature and also a few other actinobacteria. So this is more something seems to be specific for actinobacteria.
Okay, thank you. Then there's a question for Jörg about cyclic di-AMP and its interaction with ppGpp. So if cyclic di-AMP regulates the levels of ppGpp, how do you justify the activation of the TCA cycle? Secondly, does this has any role in sporulation in Bacillus species, as potassium concentration also impacts biofilm and sporulation machinery?
Okay, yes, this A was reported to have an impact on the onset of sporulation in response to DNA damage. But this is a field which certainly needs more research. The first question was how we justify the role of cyclic di-AMP in the control of the TCA cycle, right? Yeah, Regina?
Okay.
Yes, was it? What was the first question?
The first question was, is cyclic di-AMP, if cyclic di-AMP regulates the levels of ppGpp, how do you justify the activation of the TCA cycle?
Okay, one thing is that genes of the TCA cycle are also activated by the stringent response. For example, this very gene of the pyruvate carboxylase is subject to a positive stringent response. So if ppGpp accumulates, also the pyruvate carboxylase gene expression is stimulated. And we know if potassium gets low, then the cell really is only viable if it gets excess to other positively charged molecules. And the way to get access to such molecules is the production of positively charged amino acids. And this works via glutamate in the first part, at least, of the TCA cycle.
Okay, thank you, Jörg. Then I have a question for Kürşat. This is about SPIX and its role in redox homeostasis. And the question is, have you looked into the levels of glutathione and other essential sulfur metabolites that maintain redox homeostasis in response to SPIX? We can't hear you, Kürşat. Switch on.
We did not look at glutathione directly, but we looked at thioredoxines, and they are upregulated, and they're really important then also for the repair, because there's an important thing that cysteines, which get oxidized, can also sterilize aggregates. And we also have, I think, a nice experiment where we grew the cells anaerobically and heat shock them. And when you heat shock anaerobically, you have much less aggregates. So the oxidation is another important part of the stabilization and formation of aggregates, and therefore the whole SPX pathway, which is also thioredoxine and also the disulfide metabolism, is quite important for that.
Okay, thank you. And another question for Natalia. In the context of osmotic stress, what specific changes in gene expression or cellular processes are modulated by cyclic di-AMP signaling in Streptomyces? So this is about the targets, effector target system and output.
And the osmotic stress conditions?
Yeah.
Okay.
Or
So,
in the context of osmotic stress.
Well, so we did some RNA-seq experiments, I mean, harvested cells from these plates, and there are many different genes that are differentially expressed, including, for example, ACT2 in biosynthesis genes, which is an osmoprotectant, for example. But I think it is also relevant how proteins are affected on protein level, on activity level. And Streptomyces have many different ion transporters, so really a big set. And we are trying to understand how all this fits together within the cyclic di-AMP signaling network. But definitely osmoprotectant systems are also affected. And we will look into this in more detail in future.
Mm-hmm.
I think it was a good question about signals that activate the enzymes, Regina.
Oh yeah, that was a very general question actually.
Yes,
for all
you
the
can answer.
So this is difficult to answer. So basically all these or most of these enzymes that make a certain make or degrade a certain second messenger have sensory input domains. So they sense different things. And you name it. I mean, it can be almost anything that can be sensed, different molecules. It can be light, it can be membrane tension, it can be another protein, a specific state of another protein. So it's a whole slew of possible inputs. Yeah. So it's hard to give that question to someone.
Yeah, I think actually Jörg had a nice example with the star A, star B, the interaction of the two pathways, and that it's protein-protein interaction then in the end between REL and this new protein, which can sense the other metabolite. So.
Mm-hmm. Yeah, but that's more of the output, for the output. The question was more about the sensory input. Yeah?
That's even more difficult.
Yeah, yeah. And that's actually in many cases, it's the main thing that we don't know about these enzymes, what they actually sense, what they respond to. That's where a lot of research is actually going on. And then there's also another question that I found here in the chat. And this is actually, so Jörg mentioned that cyclic di-AMP under specific conditions in specific cases can be extracellular, seen from the perspective of the bacterium. And now there's a question where somebody asked, can you ever find cyclic di-GMP also outside the cell? Natalia, do you know of any example? Because I don't know of any.
Well, if I remember correctly, there was this dictyostelium case, right? Which has this transporter that can, I mean, the very low eukaryotic system that produces c-di-GMP and can actually export it. But otherwise, I think the c-di-GMP transporters, I mean, this is We don't know.
Yeah, no.
And the
Yeah.
molecule also cannot diffuse through the membrane, right? Due to the, because it's negatively charged.
Structure and size, and so it would not go spontaneously through the membranes, much less across two membranes in a gram-negative. So maybe you can comment on the cyclic di-AMP case.
Well, there's just one little thing that I would like to add. So in fact, cyclic di-AMP can be excreted, and it is excreted by pathogens.
Mhm.
And as I mentioned, it's recognized by a protein of our immune system. This protein is called STING. And what I really find extremely amazing is that we got this STING proteins from bacteria. So originally, this was kind of designed as a protein that bacteria use to fight against phage infection. And we adapted this protein, the STING protein, to sense bacterial infections, to sense cyclic di-AMP. And in fact, it can even sense cyclic di-GMP.
Yes.
And use it to stimulate our immune system. So this is really something I find absolutely fascinating. And unfortunately, I didn't have the time to mention it in the talk, so I'm happy I have the chance here.
Yes. So this is really a nice example that a part of our own human immune system is a descendant of a bacterial immune system that bacteria used to immunize themselves against phages. So it's really, really amazing. Yeah. So I think we are already a bit over time. So maybe I have to close the session now. I would like to thank all four speakers for their wonderful presentations, and everybody who joined in for staying for the whole session. And for those of you who could not make it for the whole time, there will be a record of it, and it will be available via the website of microLife. So thank you very much to everybody who was involved, and also for the people who did all the technical work in the background, whom you could not see. Thank you very much. I'm happy that everything worked so nicely. And I hope to see you soon again, at least the speakers. Okay, bye-bye. Thank you.
Bye-bye, take care.